So when we talk about protozoans, we need to also mention, of course, a few cell parts. Now, you talk about any type of cell. Of course, they're very complicated, and that includes the cells that make up our body. Even the cells of protozoans have lots of different parts that are being used to make sure that cell can function efficiently within its environment. Now, we're not going to talk about all the different parts, but we're just going to focus on a few. Now, one that we cannot leave out, of course, is going to be the nucleus. The nucleus is extremely important to the protozoan because it's going to act as a communicator. So its job actually is twofold. It communicates with the different parts of the cell, so the other organelles that are found within the cell. So it basically tells them what to do. Now, of course, its second job is to participate in cell division. Right? So in other words, it's going to contain the genetic material, it's going to have the chromosomal material, it's going to have the DNA, and that's going to be essential when it comes down to that cell dividing and passing on that information to its offspring. Now, when you talk about a nucleus, you need to look at two things here. The first one is the nucleoli. Now, again, these are things that I'm guessing you probably looked at back in biology. Now, nucleoli is going to be that very dark center to the nucleus. And so if you have if you can kind of see off to the right-hand side here, if you have a cell and you have a nucleus inside of that cell, you're going to have that very dark center inside of that nucleus, and that's going to be the place where ribosomes are going to be produced. Now, ribosomes are important because ribosomes help us to produce protein, and of course proteins are important to all cells. Plus, we can also at times now use these different types of nucleoli, because they do vary between protozoans, to actually identify the type of protozoan that you're working with. Now, we're also going to talk about something called a macronuclei, and actually you're going to talk also about a micronuclei, but we're going to focus on that again a little bit later. These tend to be found in ciliates, and so a macronuclei, micronuclei is primarily found in one group. Now, if you're talking about a macronuclei, you're talking about one that is very, very condensed. Now, it's condensed due to the chromatin that is found within that macronuclei. Now, the macronuclei in ciliates, again, as we said before, there are two different types, macro and micro. The macronuclei tends to be the one that's going to communicate, so it's going to tell the various cell parts what to do. The micronuclei is going to be the one that's actually going to participate in cell division. So this is kind of interesting because we actually have a nucleus that has two different types of subnuclei that do different things. So it sort of delineates the job in this case. Now, we also talk about mitochondria, and mitochondria are used in energy production, so they produce the ATP that is necessary to make sure that the organism can function. We have something called a Golgi apparatus, which is going to work in tandem with the endoplasmic reticulum, and its function is to basically transport and secrete certain things within the cell. And then we have, of course, plastids. Now, when we talked a little bit earlier, we had talked about two different groups of protists. We talked about the animal-like and the plant-like protists. Plastids are primarily going to be found in the plant-like protists, all right? Now, a chloroplast is one of those plastids where it's going to primarily function to take in the sunlight, to take in the energy from the sunlight. And it's going to use that energy to convert various materials into sort of a chemical type of energy. So plastids are going to be primarily lumped into that sort of plant-like category. But like we said before, Euglena was a good example where you have an organism that can basically bounce back and forth between different modes of gaining nutrition. You know, as we said before, if it's in an environment where there's no sunlight, it's going to become heterotrophic. It's going to consume other organisms. If it's in an environment that has lots of sunlight, it's going to become autotrophic, and it's going to take advantage of its chloroplasts and take in that energy and make its own food. So again, these chloroplasts, or these plastids, can be found in some of the animal-like protists. All right, so let's go ahead, and this next slide I just threw in here because I wanted to illustrate some of the parts that we had just talked about. And if you notice, let's go ahead and look maybe on the right-hand side here. You'll notice that we have, this is a euglena, by the way, so this is the one that we were talking about that can kind of bump back and forth between the different modes of gaining nutrition. We have the nucleus located right up here. In fact, if you look really closely, you can kind of see the nucleolus right there. Over here, you can notice we have a flagellum, which we had talked about, so this is considered a flagellate. That's how it's going to move throughout its environment. Now, the chloroplast, these are the green things that you see throughout the cell. These are the ones where, if there's lots of sunlight, they're going to be able to take in the light energy and convert that into food. Now on the left-hand side here, it seems as though we have sort of an amoeba-like creature. Excuse me. Again, the nucleus is located right here. Nucleolus, again, that dark area that you see right here. The plasma membrane is going to be located here. The mitochondrion, which is one that's really important because it's going to help to basically, convert that food material into usable energy. So it's going to convert it into ATP. We did mention the endoplasmic reticulum, which you can sort of see right here. And I did notice that the Golgi apparatus isn't represented, but if it was, it does tend to be in rather close proximity to the ER or the endoplasmic reticulum. And remember, the Golgi apparatus is simply there to transport secreted materials either within the cell or outside of the cell. So looking back at, again, nutrition, we had talked about there basically being two different types of nutritional processes that protozoans can utilize when it comes down to their environment. And the first one was phagocytosis. Now again, this is a holozoic type of nutrition method. So phagocytosis, this is one where it actually involves an infolding or invagination of the cell membrane. So what that means is that we're going to get the formation of something called a food vacuole. Now the easiest way to understand this is to look on the right hand side. You're going to notice that we have an amoeba, and it's an amoeba of course because you can definitely see the pseudopods on the surface of the organism. And you're going to notice we have a food particle right here. All right. Now these pseudopods are extending themselves. Now they're basically giving the food particle a big hug is what it's doing. Now as it does that, these pseudopods are actually going to come together. And when they come together, there's going to be a fusion between these two pseudopods. And then we're going to get the formation of something called a food vacuole, which is simply a pocket that's going to contain that food particle. Now once that pocket has been formed, then what the organism is going to do is it's going to secrete digestive enzymes, it's going to digest that food particle, and anything that is left will be actually taken back to the surface of the cell. That surface is going to open up and it's going to be expelled from the cell. So it's kind of the reverse of how it took in the food particle. So that's considered phagocytosis, and again, it's a holozoic type of nutrition. Now we actually have some protozoans out there that have what we would consider sort of a rudimentary mouth and even a rudimentary anus. All right. So we have this primarily is going to be found in the ciliates. We have something called a cytostome in ciliates, and this is likened to the mouth. For us, it's used to bring in these food particles, and then of course once they're brought in, we actually do still have food vacuoles that will form. Once that food has been digested and anything that can't be used needs to be expelled, they have something called a cytopage or a cytoproct, and this again is likened to the anus, and this is going to be used to expel any of that unneeded waste material. Now when it comes down to excretion, as I said before, there are some organisms out there that actually do have special ways of getting rid of their waste material, but in most cases when it comes down to excretion of metabolic waste, it's simply going to be accomplished through diffusion. And the main product, end product, in this case is going to be something called ammonia. Now in addition to excretion, we also need to remember that these organisms are found in a very fluid or very liquid environment a lot of times. And so they need to have a way to sort of maintain a fluid balance between the inside and the outside of the cell. So a lot of them will employ something called a contractile vacuole. And if you look on the right-hand side, this right here is going to be the contractile vacuole. So this is a blown-up version of this right here, and this is a paramecium. Now this is a ciliate because you can see the cilia on the outside. So what happens is that as this organism takes in fluid, it needs a way to be able to release any excess fluid. And so as this vacuole fills up with fluid, it's going to contract, it's going to squeeze really hard. And as it does that, that fluid is going to leave through this excretory pore right here. Now this is something that's going to happen many times throughout the day for these protozoans. It's going to fill up, it's going to squeeze, it's going to release the fluid. It's going to fill up, it's going to squeeze, it's going to release the fluid. And what I often tell my kids is that you need to think about it sort of like the bladder of the protozoan. As your bladder fills up, maybe you've drinking a lot of water throughout the day. As your bladder fills up, of course, you need to go to the bathroom. Once you go to the bathroom, of course, you're going to empty your bladder. Your bladder is going to shrink. And this happens periodically throughout the day. So it's sort of like the bladder of the protozoan. But its main function is to regulate, again, water or fluid within the protozoan. So looking at reproduction with protozoan, protozoans can actually reproduce in a couple of different ways. They have an asexual type of reproduction. They also have a sexual type of reproduction. So this first slide is going to focus on an asexual mode of reproducing. So if you notice, we have 3 different modes of reproduction asexually for these organisms. We have something called binary fission, budding, and multiple fission. Now, binary fission is really easy to understand because it simply means we're taking that one cell and splitting it into 2. All right, so basically that membrane, that cell membrane, is going to pinch. There's going to be replication of that genetic material within the cell. So we have 2 sets of material, one on each side. It's going to go through cytokinesis, which is a pinching of the cytoplasm, and it's going to pinch into 2 separate cells. So that's binary fission. Bi means 2. Budding, on the other hand, is similar to binary fission, but it's not an equal distribution of the cytoplasm. So we might have a protozoan like this one right here, where only a small part of the protozoan is actually going to expand and get bigger. And once it does that, it's going to again pinch like it had done before, but the distribution of cytoplasm is very unequal. Lots of cytoplasm here, not as much here. Now it still has the genetic material, the DNA has still been replicated, it still maybe has a few organelles that will be sent its way, but the size of the 2 is very different. Once it buds off, once it detaches from the parent, it still grows to the adult size. So it doesn't stay small, it still actually grows and becomes the normal protozoan, but that's considered budding. Now, if you have a multiple fission or a schizogony type of situation, this is where you actually have multiple divisions of the nucleus before the cell is going to divide. So if you look at the diagram below, we have this one cell and we have one nucleus. Then we have 2 nuclei. Then the nucleus divides again, not divides but replicates itself again, and now we have 4. And this can continue to happen over and over again until you actually end up with a cell that has lots of individual new nuclei. But then what happens is, instead of an equal split of cytoplasm like you see up here with binary fission, you have cytoplasm that actually splits in lots of different ways. It splits in lots of different ways because you do need to provide a little bit of cytoplasm to each of these individual new cells that you see here. So this is considered multiple fission. Now, if you have fission that is preceded by the union of gametes, and gametes simply mean sperm and egg, then this is not called schizogony, it's actually called sporogony. And so it's the same process, you still get multiple replications of that nuclei, but if you have a fusion of gametes beforehand, then you're going to give it a different name. But the outcome is exactly the same. Now that we've talked about asexual reproduction in protozoans, we need to also make sure that we mention sexual reproduction. Now, sexual reproduction is super important because it encourages genetic recombination. And if you can encourage genetic recombination, you can encourage evolution of that species. Now, gametes are used to restore what we call the diploid number of chromosomes. Now, if you think back, when we had talked about diploid and haploid in biology, diploid meant a full set of chromosomes. So for us, a diploid state would be 46. So every cell within our body, not including the reproductive cells, has a diploid number of 46. We would also mention something called a haploid number. And in our case, the haploid number would be 23. But again, the haploid number only occurs in our reproductive cells. So of course for guys it would be sperm, and for girls it would be the egg. Now, down here, if you notice, we have 2 different states. We have something called isogametes and anisogametes. Now, isogametes refers to gametes that all look alike. In other words, you can't distinguish between the 2. In anisogametes, these are 2 dissimilar gametes. So in humans, for example, there's a very distinct difference between a sperm and an egg. You can tell the difference between the 2. And in protozoans, you can actually have gametes that are produced where you can tell the difference between the 2. But again, there are some that you can't. Now, when fertilization occurs of that gamete, in other words, a gamete meets up with another gamete and they come together and fertilize, then we call that syngamy. All right, so we have another name for fertilization when we talk about protozoans. It's called syngamy. So it's simply a fertilization of an individual's gamete by another. Now, autogamy is when gamete nuclei arise by meiosis and fuse to form a zygote within the same organism. All right, and so what we have here is we have a protozoan that might produce maybe two different forms of a gamete. You can think about it sort of like a male and a female gamete, that they're still held within that protozoan cell. They come together and they actually fuse and they create the zygote. And the zygote is simply that fertilized egg. Now this is different from syngamy because we have fertilization of an individual gamete by another outside of the cell. So it's not found within that same organism. Now, there's not a lot of protozoans out there that will actually come together when it comes down to sexual reproduction, but paramecium are the exception to the rule. They will actually what we call conjugate, and so this is called conjugation, and this is the exchange of gametic nuclei between paired organisms, and they call them conjugants. So on the right-hand side here, you're going to notice we have an example of conjugation. So right in this area right here is when you have that exchange of gametes. And then, of course, fertilization is going to occur within the cell, and we'll have those zygotes that are going to form. So some protozoans will actually do sort of a rudimentary type of mating behavior between the two, but we call it conjugation. So the very last thing we need to look at here is we need to look at the difference between Insystem and Accessment. There are situations in the environment where some protozoans will need to protect themselves, and the way that they would protect themselves would be the formation of cysts. And a cyst is simply a hard outer covering that is produced by the Golgi apparatus within the cell. Now the Golgi apparatus is that particular cell part that is going to be used to secrete certain substances within the cell and possibly even transport those substances. And in this case, because of what is going on outside of the cell, what is indicated here is that the cell needs to produce some sort of protective barrier to protect that protozoan from the harsh environment. So it's going to produce the cyst. So that's going to be the responsibility of the Golgi apparatus. Now, again, in addition to harsh environments, there are times where there may be a situation in the protozoan's life cycle where a cyst will actually need to be produced. So this would be another situation where a cyst would be necessary. Now, once that condition becomes more favorable, in other words, it's not as harsh, or once the protozoan is at a certain part in its life cycle, it's going to perform Accessment. And again, Accessment is simply the leaving of the cyst. Now, if you look on the right-hand side here, I have an example of, I think it's a didinium, that will show you what occurs during Accessment. So I'm going to go ahead and hit this video and just kind of narrate what's going on. So again, if you notice right here, this is going to be the protozoan, and you can kind of see the protozoan moving within the cyst itself. And the cyst can be identified by this barrier that you see right out here. And so you can see that this is going to be probably a ciliate of some sort. You can see the cilia beating on this protozoan. I'm going to fast forward this just a little bit. Kind of gives us a better idea as to what's going on. So the protozoan itself is going to start moving around a little bit more, which is going to start to interfere with the integrity of the cyst itself. So basically the protozoan is going to be beginning to break through the cyst. And as we kind of skip ahead maybe a little bit further, you're going to notice on the left-hand side that the protozoan is starting to break through the cyst itself, and eventually it's going to make its way through. And so this would be considered Accessment, and basically a release from that cyst condition. So that's going to wrap up this particular screencast on Chapter 11. Please make sure that you complete the screencast form that is connected to this screencast, and I will see you next time.